All right, so we'll start off with the monomer and learn a bit about them before we go to the peptides and proteins. So amino acids, what are they? Well, the name is pretty self-implied or explanatory. Amine, it's because it's got an amine there. An acid, an organic acid, is generally a carboxylic acid, right? So it's got both an amine and a carboxylic acid. So it goes like this: carboxylic acid over here. Generally, the amine is drawn as pointing down here. And then there's some type of R group, right? So again, here's the amine, here's the carboxylic acid, and then here is the R group, and the R group is different for every amino acid. Call them AA for amino acid. Generally, these are also sometimes called alpha amino acids. And that's because this is the alpha position, right? And you can see that the amine is pointing off the alpha position. There are a lot of different amino acids found in nature, but there's about 20 that are abundant. And then, of course, I've copy pasted this image from your textbook, right? That shows you the 20 different amino acids. So some things you might notice, you notice how all of these are wedges, right? We don't see any dashes with the amine. And so similar to our sugars, the only amino acids that are abundantly found are one enantiomer. In this case, they're the L enantiomer, except not really. It's not actually the L amino acid. They just call it the L amino acid because if you draw the Fisher projection, it looks like an L sugar. So it's not, it may or may not be L depending on the amino acid, but we call them all amino acids, right? So these are the ones that are predominantly found. If you look at the stereochemistry, they're all in the S configuration. There's no ones that are in the R configuration. And then glycine has two H's, so that one is not even chiral to begin with. When you take biochem, you'll probably have to memorize all of these. We're not going to bother doing that, right? It's an organic chemistry course. We're just going to take a look at some of these, right? And so you can see that's organized, based on their properties. So first off, you have the name of the amino acid itself with the R group in green. So glycine, for example, just has an H as the R group. Alanine has just a methyl of the R group. And then the 3-letter abbreviation and then the single letter abbreviation, so lots to memorize there. And then you can see it's grouped based on their properties. So these are the amino acids with non-polar side chains, right? So they're probably going to point inward towards the protein and not away from the water. And you can see you can have like a hydrogen, a methyl, and just different types of alkyl chains, right? They're just different alkyl chains, different branching. We have a sulfur in this one. But there's plenty of other carbons, so not very polar. This one's proline's pretty cool because it's cyclic with that amine. It's interconnected there, right? Benzyl group there. And then we've got this group right there. Again, we do have a heteroatom here, but overall very nonpolar. Then over here with polar side chains, right, you can see you have an amide, another amide, an alcohol, phenol, and then a thiol, right? Things that are polar. And then acidic side chains, right? So in this case, these amino acids are more acidic than your average amino acid. And as usual in organic chemistry, the acids are carboxylic acids. And then amino acids with basic side chains. These are generally nitrogen containing ones, right? So you can see this one's real cool with these 3 nitrogens, pretty cool structure there. We've got histidine right there. Again, pretty cool structure. Lysine, not as cool, but still interesting. So lots of amino acids there. Again, don't have to memorize them, but you should be able to look at it and, think about the properties and whatnot.